Hi students, welcome to Nurse Educatrice video series in microbiology for nurses. So in the previous video, we discussed about the basics of microbiology, importance of microbiology in nursing and the historical aspect of microbiology. So in the coming up video, we will be discussing about the anatomical structure and classification of microorganism. So what is microorganism? And the microorganisms are heterogeneous group of organisms which are classified under the kingdom called Protista. Like plants, animals, there is a kingdom called Protista where the microorganisms are coming under this kingdom. So this Protista is again divided into prokaryotes and eukaryotes. Bacteria comes under prokaryotes and fungi, molds and protozoa comes under eukaryotes. So what is the difference between prokaryotes and eukaryotes? And prokaryotes has a different types of cell in which there is nucleoid and there is a capsule. There are appendages like flagellum and fimbriae, and there is a cell membrane and a cell wall. Whereas eukaryotes, they have nucleus and nucleolus, and the mitochondria and endoplasmic reticulum, they don't have the outer cell wall and they don't have the appendages like flagellum or fimbriae. So bacteria comes under prokaryotes. What is the size of microorganisms? The microorganism size or the unit for measuring a microorganism is called microns. So 1 micron, that is mu, is 1,000th of millimeter. 1 millimicron or a nanometer is 1,000th of the micron. So generally the bacteria's size is represented in microns. 1 micron is 1,000th of millimeter. 1 millimicron or a nanometer is 1,000th of the micron. So if we talk about the size of the bacteria, most of the bacteria usually measures 0.2 to 1.5 microns in diameter and 3 to 5 microns in length. So we need microscope to visualize these microorganisms. So most of the medical important bacteria comes under 0.2 to 1.5 micron diameter and 3 to 5 microns in length. Shapes of bacteria. The bacteria shapes are of 3 main types, that is cocci, bacilli and spiral. Cocci is nothing but spherical shaped bacteria. They stay single or they stay together like diplococci, or they stay in group like staphylococci, or they arrange in a linear pattern. The spheres or the cocci is arranged in linear patterns in case of streptococci. So the arrangement of the cocci is in different bacteria or different unique patterns. So the spherical shaped bacteria are called cocci. So diplococci is Streptococcus pneumoniae. You can see there are 2 spherical shapes here, and in the staphylococci they are group of spherical shapes together, staph aureus. And then streptococci pyogenes, they arrange in a linear pattern in a linear sequence. So rod shaped bacteria are called bacilli. So they will be as a single rod, or they may arrange in the linear sequence to form a chain of bacilli. And the next is spiral shapes. Spiral shaped bacteria, the best example is Vibrio cholerae. It is a spiral shaped bacteria. And Helicobacter pylori, which has a spiral shape, and the spirochetes, Treponema pallidum. So spiral shape is again divided as normal spiral, spirilla and spirochetes. You can see the shape, difference of the shapes in this picture. Okay, so bacteria, according to the shapes, they are classified as cocci, bacilli and spirals. What is the anatomy of bacterial cell? Here I am not going to teach you a complete anatomy of bacteria. I will be focusing on the exam point of view. So you should know how to draw an anatomical structure of bacterial cell, which we have studied basics in our schools. So it has a cell wall, a capsule, cytoplasm, plasma membrane. So these are all the normal anatomical cell structure which we studied earlier even in anatomy human cell. So it has some additional things like flagella and fimbriae as appendages. And if we see what is the bacterial cell wall, the importance given here, because the classification of one type of bacteria is according to the cell wall arrangement. That is gram-positive cocci and gram-negative cocci. The cell wall is outermost layer of the cell, and the main function of the cell wall here in bacteria is to protect the cell, maintain the shape of the cell, and it plays a vital role in cell division. It assists in attachment of bacteria to the host cell, presence of surface appendages like flagella and fimbriae. So the bacterial classification according to the cell wall, it is divided into or classified into gram-positive and gram-negative cocci. So what is the difference here? From the picture itself you can see the peptidoglycan layer in gram-positive is very thick. The green thing which I have noted here as peptidoglycan, which is very thick in gram-positive, whereas in gram-negative it is a very thin layer. Okay, and gram-positive has around 80 mm thickness of peptidoglycan, whereas in gram-negative it is of 5 to 10 mm thick of the peptidoglycan. This peptidoglycan is covered by periplasmic space in gram-negative bacteria, whereas in gram-positive there is no periplasmic space. That is the yellow color in which I have given here in gram-negative cocci. And this gram-negative cocci has an outer membrane, which is the red part which is covering the periplasmic space, whereas in gram-positive there is no outer membrane. Usually when we do a gram stain, we can differentiate gram-positive and gram-negative by the color of the stain. Okay, whereas gram-positive gives a purple color, whereas gram-negative gives a pink color. That we will discuss in the gram staining technique in detail. So these are all the difference between gram-positive and gram-negative cell wall. It is a very frequently asked university question. So you can draw this picture and you can differentiate so easily so that you can gain marks. Here this is the diagrammatic representation of differentiation of gram-positive and gram-negative cell wall. Gram-positive bacteria retain the crystal violet stain, and whereas gram-negative, as I said, it retains the crystal violet stain during the and do not retain the crystal violet. It appears purple color, it appears pink color. The outer membrane is present in gram-positive, whereas in gram-negative there is no outer membrane. And peptidoglycan layer is thick and multi-layered, and whereas peptidoglycan layer is single layered in the gram-negative cocci. Periplasmic space is absent, whereas periplasmic space is present in the gram-negative. Cell wall is around 20 to 80 mm thickness, as we have seen the green color layer which is thick, whereas in gram-negative it is thin of 5 to 10 mm. The cell wall is smooth, whereas cell wall is wavy in the gram-negative. The cell wall contains virtually non-lipopolysaccharide content, whereas here it has a high lipopolysaccharide content. Some of the examples of gram-positive or Acetobacillus, Actinomyces, Bacillus streptococci and Clostridium corynebacterium. The examples of gram-negative organisms are Acinetobacter, Chlamydia, Borrelia, Bordetella. So these are all some of the examples of gram-negative and gram-positive cocci. Next we will move on to important appendages present in the bacteria, that is flagella and fimbriae. Flagella is a long whip-like structure which is present in some bacteria. This is the long thing which I have marked here as flagellum, is the whip-like structure which is present in the bacteria. So what is the use of this flagella? It helps the bacteria in locomotion. In the movement of bacteria, the flagella helps. And also it acts like a sensory organs which sense the temperature change and the chemical changes happening in the external environment of the bacterial cell. So it acts like a sensory organ as well as it takes part in the locomotion of bacteria. The next is fimbriae. Fimbriae are bristles like short fibers occurs outer surface of some gram positive and gram negative bacteria. It helps the bacteria to stick to the host cell surface. You can see this small red thin lines which I have drawn here in the bacterial cell wall, which is called fimbriae. What is the use of fimbriae? It helps the bacteria to stick to the surface of the host cell. So there is a university question which is like differentiate between flagella and fimbria, which is a very commonly asked question and repeated twice or thrice in the university question papers. So you should know in the anatomy of the bacteria, the main important thing is cell wall, because the bacteria is classified according to the cell wall as gram positive and gram negative. And the other appendages like flagella and fimbria you should focus. Next we will talk about the bacterial nutrient. Why? Because we want the bacteria to grow in the labs and we should know what all the things are needed. And in case of, if we understand what are the things needed for the bacterial growth, we can suppress by reducing the bacterial infection. So we should know what are all the nutrients the bacteria needs to grow. The minimum nutritional requirements. The first thing is moisture. So wherever there is a moisture, there is risk of bacterial growth. That's why whenever we talk to the patient regarding the hygiene, you should talk about this moisture. Do not leave moisture in any places, especially in between the toes or in between the fingers where the bacteria may sit and grow. Source of carbon or carbon dioxide is necessary for the bacteria. Source of nitrogen, some inorganic salts, and there are very minimal bacterial vitamins, and the pH for the bacteria to grow is 7.2 to 7.6. So in this, most of the bacteria in this pH range, they will grow faster. Light. Regarding the light, bacteria needs darkness to grow. So whenever there is a dark room, there is high chance of bacteria. So you should talk, when you talk to the patient, you should talk about the lighting and the air movement and the moisture. So all these things, so that the patient will be aware of what are all the things necessary for the bacterial growth, so they can reduce those things. Classification of bacteria. So according to the nutrients, preparation of the nutrition, the bacteria are classified as phototrophs, chemotrophs, autotrophs and heterotrophs. So from the term itself, you all know that what is phototroph? If the bacteria obtain energy from the sunlight, they are called phototrophic bacteria. Chemotrophic bacteria, they obtain energy from the chemical reactions. What is autotrophs? They synthesize their own organic compounds by themselves. They themselves prepare their nutrition, which is called autotrophs. What is heterotrophs? They are unable to synthesize their own things, so they usually depend on some other organism for the organic compounds. They are called heterotrophs. So according to the nutrients, the bacteria is classified as phototrophs, chemotrophs, autotrophs and heterotrophs. According to the metabolism, the bacteria is classified as aerobic bacteria, anaerobic bacteria, microaerophilic bacteria. So what is aerobic bacteria? Aerobic, it means need oxygen for growth. So if the bacteria depends on the oxygen, we say it as aerobic bacteria. If the bacteria grow in the absence of oxygen, we say it as anaerobic bacteria. The best example is Clostridium tetani. So that's why whenever we get injured in any of the iron rods or stones or anywhere, we are worried about the Clostridium tetani and we take tetanus toxoid injection. So anaerobic bacteria grow in the absence of oxygen. The next one is microaerophilic bacteria. So they grow in the presence of very low oxygen. There are some bacteria which grow in the presence of very minimal oxygen. They are called microaerophilic bacteria. So according to the metabolism, the bacteria is classified as aerobic, anaerobic and microaerophilic bacteria. According to temperature. So according to the temperature where they need for their growth, the best optimum temperature for most of the bacteria is 37 degrees Celsius. So if the room temperature is too high, what happens? The bacteria will grow faster. So mesophilic bacteria are the bacteria which grow in 25 to 40 degree Celsius. So 25 to 40 degrees Celsius in the temperature, if the bacteria survive, they are called mesophilic bacteria. Psychrophilic bacteria which grows at the extreme low temperatures like below 20 degrees Celsius, and even some may grow in minus 7 degrees Celsius. Especially there are some bacteria which grow in the items which we keep in the fridge. They are called psychrophilic bacteria, like minus 7, minus 2, minus 3 degrees Celsius. Also there are few bacteria which grow. So you have to take care of the items when we keep in the fridge. Check whether do they have a bacterial growth, because sometimes it happens with psychrophilic bacteria. The next one is thermophilic bacteria which grows at extreme temperatures, hot temperatures like 55 to 80 degrees Celsius. Where the problem comes here is sterilization and steaming may not affect this thermophilic bacteria. So you should always remember 100 degree is minimum essential. But some bacteria even can grow at 250 degree Celsius. So thermophilic bacteria which tolerates extreme high temperature like 55 to 80. Psychrophilic bacteria which grows best at a temperature of less than 20 degrees Celsius, and mesophilic bacteria needs temperature of like 25 to 40 degrees Celsius. So at 37 degrees Celsius is a very optimum temperature for the bacteria to grow effectively. So when you make them grow in the lab for the culture, of course you should maintain like 30 to 40 degrees Celsius for the bacteria to grow. The next we talk about bacterial spore here, because this is one of the repeated question from the university, and also it is the spores are the toughest organism which is difficult to destroy. So as a nurse we should know what is spore. Microorganisms sense and adapt to changes in their environment. So when some changes happens, the microorganism also change to live long. Where if they want to live longer, they also sense and adapt to their changes. When favored nutrients are exhausted, the bacterium produces a dormant and highly resistant cell to preserve cell's genetic material in times of extreme stress. So what happens when there is no nutrient supply and when there is no food, the bacteria will go into a dormant stage, but just to protect their nucleic or genetic materials. So what they do is they just become spore and they survive in the extremely high stress. So this is called bacterial spore. So what happens when the microorganisms, they don't have nutrients or they are not getting enough food for their metabolism, what they do, they don't die. So just to prevent the death of the bacterial cell, they move to dormant stage called spores. So what they do, just they wrap up the nucleic material which is inside the cell, and they stay safe in extreme stress also. They are called endospores, bacterial spores. So they survive environmental assaults that would normally kill the bacterium. So usually what happens when we boil or in the extreme high temperature, what happens, the bacteria die. Yes, so to prevent this, some of the bacteria, they change into spores, okay, or endospores, just to prevent the death and survive in the difficult situation. These stresses include like high temperature, high UV radiation, chemical damage, enzymatic destruction. These all will not do anything with the endospores. So what is the problem here is, for us it is going to be very difficult to kill these spores. So some of the species like Bacillus and Clostridium, they have a capacity of the bacteria to change into a dormant stage called spores. Clear? So what is a spore? During there is a change in the environment, when there is no food available, what this bacteria do, they convert themselves into a spores, which is a dormant stage where they protect their nucleic materials and they live in their extreme difficult situation. This is one good thing as a human being we should always remember. When there is difficulty happens, you should behave like a bacterial spores. Some of the species are Bacillus and Clostridium, which has a capacity of moving them into a endospores. So they cannot be, these spores or they cannot be killed by high temperature or high UV radiation or any chemical change, enzymatic destruction. Nothing will do any change in the bacterial spore. So what happens when they get a nutrients, they will start themselves to become a bacteria and they will survive again in the environment. So when there is no food, they convert themselves into endospores, and when there is nutrients available, they convert themselves into bacteria. So this is the structure of the spore. So what happens, they just wrap up into one small teeny tiny hole, okay, just to protect the nucleic material. That's why it looks round, the inner membrane, and there is a cortex, and there is a outer membrane and a coat which protects the inner core of the bacteria. The next is bacterial growth curve. This is very, very important from the microbiology aspect, because when we make the bacteria to grow in a culture plate for the study, you should know how the bacterial growth phase takes place. You all know generally the growth happens through binary fission and it happens in phases. So you should know in a culture plate, in a lab, how the bacterial growth happens. So it happens in 4 phases. The first phase is called lag phase. So for example, imagine that you are taking a culture plate and you have added all the nutrients for the bacteria to grow. We have discussed like what temperature they grow, what pH they grow, what are the things they need for the growth. All those things we have already discussed. So you put a bacteria in a culture plate. So it starts taking the nutrition, and in the lag phase, what happens is like 1 to 4 hours, it's an adaptation phase. You just drop a bacteria inside a culture plate. It just stays there for 1 to 4 hours just to adapt to the new situation. And the next phase is log phase, where the growth, it will observe what are all the nutrients there, what is the food available there, how is the temperature, okay, cool, I can grow. Then it will start to multiply. So the growth doubles in 8 hours. That is called log phase. You can see here the blue phase, which is growing. Yes, the bacteria starts multiplying. So the graph goes up, which is called log phase. The next is stationary phase. Here what happens, it went up to the maximum growth and there is a limitation of the growth because the nutrients are depleting. Like nutrients are reducing because they used all the nutrients whatever is available. So they just stay there stationary. So they don't grow, they don't die also. So just stay there, which is called stationary phase. It is like hours to days they will stay in the stationary phase. Here you can see in the picture the green one, Where there is no destruction, there is no growth. The bacteria stays in a same place. And then the next phase is decline phase, where there is no nutrients available because the bacteria used all the nutrients available in that plate, culture plate. Exhaustion of nutrients happens, so the death of cells happens. It also takes like hours to days. Here you can see the pink curve which comes down is death phase or decline phase. So this is how the growth curve or a cycle of bacteria happens, especially in vitro, in the lab. Okay, so what happens? The first thing is lag phase. Lag phase means it stays there waiting for the change. So you put a bacteria inside the culture plate, it is absorbing its environmental change and it is waiting. And it starts up to take the nutrients where they become multiply and grow. That is called log phase or growth phase. And at certain point what happens, their nutrition limitation happens and they just stay in the stationary phase where there is no growth or no death happens. And in the decline phase what happens, the nutrient depletion happens, there is no food, nothing, so the bacteria will start to die. Okay, but the count of the cells will be same. Okay, so this is the growth curve which happens in bacteria, which is called bacterial growth curve, which has been asked twice in your university question paper. So there are 4 phases in bacterial growth curve happens: lag phase, log phase, stationary phase and decline phase. Clear? Hope you understood. We have covered in this video the anatomical aspect of bacteria, especially the exam point of view. What is the gram-positive and gram-negative cocci, and what is flagella, what is fimbriae, and the different classification of bacteria according to the nutrients, according to the metabolism, according to the temperature. And we have discussed about bacterial spores and bacterial growth curve. See you in next video with some other content. Thank you.